Yeah, I think one of the really important things about the system is that you've got this interaction between this ant and this fungus. The ants cut the leaves, they bring it back to their colonies, and they use it. They don't eat the leaves themselves, but they actually use it to grow this fungus. And this fungus supports the entire colony. And this is very reminiscent, very similar to what we do as humans in terms of our ability to grow food. And arguably, it's one of the most important aspects of what gave rise to modern human civilization. These ants have been doing this for roughly about 50 million years, and the system is really evolved to really support the growth of these ants and also support the growth of the fungus itself.
The research we do is really heavily focused on leaf-cutting ants, and this is a fascinating system from a biological perspective. The system has the amazing behavior of the ants that cut leaves, as you know, Garret, seeing them in the tropics. It's spectacular to watch them cutting leaves and carrying the leaves, and it's really a fun system to work on from a splashy biology kind of visual aspect. But the reason we study the ants, leaf-cutting ants, is not just because of their interesting behavior, but it's because of the complex symbioses they engage in. And so the work we do is really focused on understanding symbiotic associations between animals and microbes. And the system, leaf-cutting ant system, as you know, has a combination of animals with fungi, the fungus the ants grow, fungal pathogens in the system, and then microbial components in the sense of bacteria that produce antibiotics, bacteria that fix nitrogen to fertilize the system, and bacteria that appear to be helping break down plant biomass. And so you have this complex community of organisms that are working together in this association that really are the reason that leaf-cutting ants are so successful. And so understanding this symbiotic association, I think, is important for informing on other types of symbiotic associations, like even the human microbiome.
Yeah, I think one of the really important things about the system is that you've got this interaction between this ant and this fungus. The ants cut the leaves, they bring it back to their colonies, and they use it. They don't eat the leaves themselves, but they actually use it to grow this fungus. And this fungus supports the entire colony. And this is very reminiscent, very similar to what we do as humans in terms of our ability to grow food. And arguably, it's one of the most important aspects of what gave rise to modern human civilization. These ants have been doing this for roughly about 50 million years, and the system is really evolved to really support the growth of these ants and also support the growth of the fungus itself too. And just like in human agriculture, these ants and their fungus, they face the same problems that we do. They actually have pathogens that can come into the system, and they actually have to deal with it. And one of the most impressive ways that the ants deal with this pathogen, which is in fact another fungus, is they deal with it in ways very similar to the way we as humans deal with it. So they actually have figured out how to weed. They can tell the difference between their fungus and the pathogenic fungus, and they can actually go through and pick out the pathogenic fungus and take it outside of the colonies and throw it away. They've also developed a second approach, which is in fact antibiotic use, and very much similar to the way we employ pesticides. So they actually form this other interesting mutualism with a bacteria that they grow in their bodies, and the bacteria produces an antifungal compound, and this antifungal compound actually prevents the growth, inhibits the growth of the pathogenic fungus, and has very little effect on the fungus that they grow for food. So you've got this really fantastic interaction between pathogens and mutualists that are really helping to support this really complex symbiosis.
One of the big questions in my mind in biology is the getting at species interactions. A lot of the processes that have shaped diversity involve species interactions, mutualisms, where two organisms benefit, parasitism, where you have exploitation of one organism by another. These kinds of complex community interactions is really what I believe shapes diversity of life, in terms of species diversity, phylogenetic diversity, and even just genomic diversity and genetic diversity is these complex interactions. And as you know, Garret, teasing apart those complex interactions is very challenging. In very few systems can you actually work on them very well. And so one of the things that I'm most excited about with the leaf-cutting ant stuff moving forward is our ability to understand how these complex sets of symbiotic associations evolved and their interconnection. And sequencing is going to play a critical role in teasing this apart, and is really exciting part. So the idea that for our sequencing project of looking at coevolution of a community over evolutionary time, of this complex community is I think one of the most exciting aspects of what we're able to do with sequencing.
Sequencing technology has really proliferated over the last 10, 15 years. I mean, the Human Genome Sequencing Project was such a major advance in our lifetime, and being able to now capitalize on the technologies that have come forth from that project. And when you and I and other people were first faced with this question of how do we go about really digging deep into the question of what these ants are doing, one of the first things was to actually go through and start sequencing the genomes of these organisms to really get at the genetic underpinnings of what's going on, really trying to pinpoint what are the things that are really driving this community. Can we tell some of the changes that have actually undergone within these genomes compared to other organisms, to try and figure out what's really going on? And it was kind of interesting. We were presented with this opportunity with the Roche 10G project, and I was a postdoc in Cameron's lab at the time, and we actually had contacts with other people who are known veterans within the community of sequencing, like George Weinstock and Sandra Clifton and other folks like that. And we just sat down and said, here is our system. Here's what we really wanted to do. We really wanted to sequence every single piece and component to really get at the full community genome, if you want to call it that. And so we wrote a proposal and sent it in to Roche, who had just basically asked the question, what would you do if you had 10 gigabases worth of sequencing, right? And 10 gigabases worth of sequencing, in maybe more practical terms, is the equivalent of like 3 human genomes. It's a massive amount of sequencing. So what would you actually do if you had that amount of data in your hand? And that's what we proposed. We proposed, in fact, to sequence the genome of the leafcutter ant and all of its players, so the fungus it grows, the pathogen that attacks the fungus, the bacteria that grows on its body, and we even proposed sequencing all of the microbes that live inside of this fungus garden and interact with all the players in the system. And so we submitted this to Roche, and we were very fortunate, I think, to get to be funded through this. In fact, if I remember correctly, there was only 2 winners of this, ours and another group out in Europe. We just kind of embarked on this journey of the first thing we wanted to do was sequence the leafcutter ant genome. And so,
Yeah, and I think one of the things that was critical to the success of the project was the long reads, because the challenge of getting at these unique communities of organisms away from model organisms, to get at species interactions is really largely about de novo genome sequencing, and,
right.
and the long reads were critical for us in terms of making progress on our integration of genomics into our biology.
Because at that time, we actually did not have an ant genome sequenced at all. So it's really difficult to sequence something from scratch when you don't have anything to compare it to, or even something that's really close. I think the closest at that time was the bee genome. And the honeybee genome is quite different, as we now realize, from the ant genome. And so really having those long reads really facilitated putting together this genome. In fact, that was what the Human Genome Project was all about. When they first started the Human Genome Project, they had these 800 base pairs worth of reads that they were able to use to assemble into the first human genome. And so, when we kind of approached this, it was one of those things where having access to these and the newer technologies such as paired-end sequencing really made a huge difference. I think one of the biggest advances for us was really sequencing a 20 KB paired-end library to kind of, as a scaffold to put our sequences onto, and that really drove up the coverage and really turned it into a really nice genome at the end of the day. So.